nervous system too, when we will discuss why the neurons can conduct the impulse along their axons. We have a cell, and if we put a voltmeter in and outside of the cell, we would be able to measure minus 70 millivolts along the membrane, that the inside is more negative to the outside. The general value is minus 60, minus 80 millivolts, and we call it membrane potential. That is the same as resting potential. Inside of the cell, we have cations, mostly potassium ions. Outside, we have very few. And outside has more sodium ions, cations, and very few inside. The balance, this uneven distribution of the cations or distribution is maintained by the sodium-potassium pump that uses ATP to move 2 potassium ions inside and kick out 3 sodium ions outside into the extracellular area. Besides the sodium-potassium pump, there are other ion channels along the membrane, and they are ungated, just only, actually before that, we have also chlorine ions outside, tons of them, very few inside, as well as negative ions, the phosphate ions, as well as proteins with negatively charged. For example, they have a carboxyl group or phosphate group, and they give the huge negative charge compared to the outside. Besides that, we have ion channels, and they are not gated. You will see it why I say not gated. Potassium want to go outside, and it's leaky potassium channels. The potassium goes down its concentration gradient, as well as we have sodium leaky channels, non-gated, and the sodium goes inside by itself. Is a passive transport driven by concentration gradient. But the membrane potential is made up by chemical potential energy that comes from the concentration change of different ions, and the electric potential energy that comes from charged particles, different charged particles along the membranes. The ATP, actually the sodium-potassium pump, contributes to the net positive ion flow to the outside. But we have also negative ions inside of the cell that contributes to be more negative. So action potential. How the signal conducted along the axon. So as we said, potassium ions inside of the cell, more of them, and sodium ions outside of the cell. We have the sodium-potassium pump. We are focusing only on the sodium and potassium, not on the chlorine and the phosphate and the proteins. So we have the leaky, ungated ion channels for the potassium and for the sodium, and we have the sodium-potassium pump that works with ATP. Now, additional to that, we have gated ion channels, which open or close in response to stimuli and alter the membrane potential. So open or close is due to different stimulus and alters the membrane potential. That is really, really the point. So the gated ion channels can be gated potassium ion channel. When it opens, the potassium goes outside and hyperpolarization would take place. The potential difference would reach to minus 90 millivolts. And also we have sodium gated ion channels. When it opens, the sodium zooms in and depolarization would happen along the membrane. It can reach up till minus 50 millivolts or even more later on. If massive change in membrane potential will take place, action potential will happen. And when action potential happens, the change of membrane potential from one place to another will happen. So the change of membrane potential starts to spread to adjacent regions. This is basically the signal conduction along the neuron, along the axon in the neuron. For example, very fast recap it, what would happen. The sodium gated channel opens because answer for a stimulus. Sodium moves in, more sodium gated channels open up, and it's kind of a positive feedback in here. The membrane potential moves towards a positive value, still negative but more positive, towards the positive, and until it reaches a threshold, so it's all positive feedback reaction, reaches a threshold, a value when all or none event happens and the action potential reaches to the top and reaches the same strength all the time. How about we just check how we can graph this action potential and explain what happens at each step. So the y-axis will be the voltage potential, voltage across the membrane. We will start with minus 70 millivolts. That is really the resting potential. We have the minus 50, that will be the threshold for us. Here comes the stimulus, so more and more sodium gated channels starts to open and the membrane starts to depolarize. More sodium gets in, so it increases its membrane potential until it reaches a point, the threshold point, when all of the sodium gated channels starts to open up. In here at the depolarization phase, some sodium gated channels open, but reaching a threshold, all of them, that really let the sodium zoom in like crazy into the cell. And at the top, like 30 or 50 millivolts positive, the sodium gated channels start to close and the potassium gated channels start to open. Down this slope, the potassium gated channels still open until, it overshoots a little bit. This is undershot of the membrane potential. Still, the potassium is zooming out because of its inertia, I bet. So at the bottom, the potassium gate starts to close and the sodium potassium pump will bring back the membrane potential to the resting potential. Now, this hyperpolarization is really important again. This is called the refractory period. At this point, the sodium gated channels cannot respond to stimulus. Why is that so important? Because the depolarization can go only in one direction. It can move forward but not backward because the sodium gated channels cannot respond at this point. Just only the forward motion will go. The speed of the action potential depends on the diameter of the nerve. The thicker the nerve, the faster it goes, as well as the presence or the absence of myelin sheath. Myelin sheath is really important. Go back to the first video. That's what you had. These oranges are the myelin sheath that I drew in the central nervous system. This is kind of insulation. It's our lipid material. In the central nervous system, we call those cells oligodendrocytes, that they do the myelin sheath. In the peripheral nervous system, they are the Schwann cells. Both of them are glial cells. They make the insulation for the axons. Why is that so good? Because only between the myelin sheath we have gated channels. These places are called the nodes of Ranvier. And these gated channels are only there, so the action, the conduction is jumping from one node of Ranvier to another. So it's a saltatorial conduction, and that's it. Very fast.